Welcome everybody to the Targeting Coronavirus entry session, and we will have two speakers. And I would like to introduce our first speaker, Lynn Conley. She's an infectious disease physician and the medical officer at Adagio Therapeutics, and PhD in cell biology from Francisco, and then completed her internal medicine residency at Stanford and her infectious disease fellowship at the University of Washington. She subsequently joined the faculty at Washington before returning to Lynn was the Senior Vice President for Clinical Research at Vir Biotechnology and Vice President and Head of Late Development at Achaogen. Today, Lynn will discuss monoclonal antibody therapies directed against SARS-CoV-2 spike protein. Lynn.
Thank you so much for that kind introduction, and I'd like to thank the organizers for the invitation to speak at this virtual event today. I'd like to start with a brief introduction to Adagio. Adagio is a privately held company currently quartered in Massachusetts that was spun out of Adimab, which is a premier antibody discovery and optimization company. We've complemented Adimab's deep domain expertise in B-cell immunology to viral pathogens with our own expertise in the development and commercialization of novel agents for both important bacterial and viral pathogens. And today, I'd like to tell you about Adagio's broadly neutralizing monoclonal antibody program to address the current SARS-CoV-2 pandemic, as well as future beta coronavirus threats with pandemic potential. From the outset, Adagio has been focused on solutions not only for the current pandemic, but also for additional members of the coronavirus family with pandemic potential, as we believe that novel coronaviruses will continue to emerge just as SARS-CoV-2 has followed SARS and MERS. We believe this because there is massive genetic diversity among coronaviruses circulating in animal reservoirs, and at least a subset of these viruses, known as the sarbecoviruses or SARS-like viruses, are able to directly infect human cells by the ACE2 receptor, allowing for direct spillover into humans. Notably, as shown on the graph on the left, many of these SARS-like viruses are circulating in the same regions, offering ample opportunity for recombination and generation of novel viruses. For example, studies have shown that up to 20% of bats in China harbor coronaviruses, and importantly, a substantial portion of residents in rural China show serological evidence of exposure to these SARS-related bat viruses. This tells us that these viruses are spilling over from bats into humans relatively frequently, increasing the probability we will see the emergence of another virus in the human population that has struck the right balance between pathogenicity and transmissibility. The current SARS-CoV-2 pandemic has also been characterized by tremendous genetic diversity, with the global emergence of thousands of viral variants with changes in the spike protein, which is the key viral determinant of viral entry. Some of these variants have emerged with higher frequency than others, likely due to a fitness advantage. Examples of such variants include B117 or alpha, B1351, also known as beta, and P1, also known as a gamma variant, which were first detected in the UK, South Africa, and Brazil, respectively. This paper, published in The Lancet, illustrates the emergence of the P1 lineage in Manaus, Brazil. Based on serological testing of blood donors, it was estimated that approximately 76% of the population in this city had been infected with SARS-CoV-2 by October of 2020, after initial surge of cases in the spring of that year. However, in this context of high seroprevalence, a second surge of COVID-19 cases was observed, occurring beginning in January of 2021, which coincided with the emergence of the P1 lineage. This variant contains several mutations in the spike protein, leading to speculation that these changes may have conferred fitness advantages to this variant, such as higher inherent transmissibility and the ability to evade neutralizing antibody responses. Multiple studies have now shown that many of these variants, and most notably the P1 and B1351 variants, are indeed relatively resistant to neutralization by antibodies induced by both COVID-19 vaccines and natural infection. Some of that data is shown here. The figure on the left shows that the B1351 variant in particular is very resistant to neutralization by vaccine-induced antibodies, with an approximately 30 to 40-fold decrease in neutralization potency relative to wild-type virus for sera from vaccine recipients. Notably, nearly 40% of vaccine recipients included in this study did not have detectable neutralization of at least one of the B1351 variants tested. As shown on the right, similar results have been generated for convalescent sera, showing that antibodies induced by natural infection are also less potent at neutralizing the B1351 and P1 variants. We and others believe that these variants have arisen due to evade immune selection pressure from neutralizing antibodies. This idea is supported by the finding that many variants share similar mutations in the receptor binding domain, or RBD, of the spike protein, which is the primary target of the neutralizing antibody response. For example, both the B1351 and P1 variants possess mutations at E484, K417, and N501 of the spike protein. The hypothesis that variants are emerging to escape immune selection pressure is further supported by the now numerous studies describing antibody responses to SARS-CoV-2. And what these studies have shown is that the antibody response to this virus is surprisingly public. What I mean by this is that many different people are making very similar types of antibodies that are not only binding to the same epitopes, but are actually engaging the same amino acid residues within these epitopes. These antibodies have been categorized into three classes, and I am highlighting here the mutations that are associated with escape for many antibodies in these classes. For example, K174N or T mutations are common escapes seen for class 1 antibodies, and E484K is a common escape mutation for class 2 antibodies. Many studies, such as the one shown here on the right from Jesse's Bloom lab, show that mutations that position E484 in particular strongly reduce neutralization by convalescent sera across multiple different donors. It is likely that these mutations are arising due to immune selection pressure exerted on the virus by these common classes of SARS-CoV-2 antibodies. Importantly, most clinical stage antibodies, including antibodies currently available under emergency use authorization in the US, were isolated from COVID-19 patients. As such, many of these antibodies belong to these common antibody classes shown here and are thus impacted by these same mutations, as I will illustrate in the next slide. The data I'm showing you here is authentic virus neutralization data generated in Gavin Screeton's lab at Oxford University for several clinical stage antibodies against an original SARS-CoV-2 variant, Victoria, and three variants of concern, B117, B1351, and P1. As you can see from these data, antibodies belonging to the common classes 1 and 2, such as Regeneron 01933, LycoV555, LycoV016, and AZD8895, show varying losses of potency against the B1351 and P1 variants. In the case of the Lilly cocktail, we see complete loss of neutralizing activity for both antibodies against these two critical variants. For this reason, the distribution of this product has recently been paused to several US states where the combined frequency of the P1 and B351 variants exceeds 10%. These data illustrate the need for broadly neutralizing options that are less prone to viral escape. This slide here shows you the evolution of SARS-CoV-2 over time, highlighting some of the reasons that I've already explained as to why we believe additional SARS-CoV variants will continue to arise, thus underscoring the need for these broadly neutralizing options that are less prone to viral escape. Oops, I went too far. Based on a large body of work previously conducted by our CSO, Laura Walker, on broadly neutralizing antibody responses to other viral pathogens, we reasoned that, at least in principle, inherent features of broadly neutralizing antibodies should offer a higher barrier to resistance. There are two main reasons for this. The first is that these antibodies tend to bind to immunorecessive epitopes. These are epitopes that are not readily targeted by endogenous immune responses, thus reducing immune selection pressure on these sites. The second, and perhaps more important reason, is that by definition, broadly neutralizing antibodies engage highly conserved residues, which are often important for viral fitness. Although escape in vitro can be generated against broadly neutralizing antibodies, the number of escape pathways is typically lower for these types of antibodies. In the next slide, I'll show you the efforts that we went through in order to identify broadly neutralizing antibodies to SARS-CoV-2. In order to do this, we first started by obtaining a blood sample from a survivor of the original 2003 SARS outbreak. We then sorted B cells from the sample using SARS-CoV-2 spike protein. Because this donor was never infected with SARS-CoV-2, by definition, we were pulling out broadly binding antibodies. From these efforts, 200 cross-binding antibodies were identified, and of these, 7 showed broadly neutralizing activity. 3 of these broadly neutralizing antibodies were chosen for affinity engineering. The goals of the affinity engineering process were twofold. 1, to improve binding affinity and neutralization potency against SARS-CoV-2, while maintaining or ideally even improving the potency against other SARS-like viruses. And 2, to retain favorable developability properties of the antibodies, such as low polyreactivity, which is associated with a favorable pharmacokinetic profile in humans, as well as low viscosity, in order to support convenient intramuscular administration for use in the clinic. Of the 3 engineered antibodies, Adagio 2 was chosen as our lead clinical candidate for clinical development, and Adagio 1 was chosen for further development as a future potential combination partner for Adagio 2. We next engineered the Fc portion of these molecules to extend the half-life, specifically choosing a modification that is not anticipated to impact innate immune effector function. In the next few slides, I will illustrate how the affinity engineering process led to significant improvement in neutralization potency without negatively impacting the breadth of activity of these antibodies. The figure on the left is showing the binding affinity evolution for Adagio 2, with the parent molecule shown in green, the progeny molecule shown in orange, and the ultimate product, Adagio 2, shown in gray. These data demonstrate a remarkable 500-fold improvement in monovalent binding affinity to the SARS-CoV-2 spike protein through 2 rounds of affinity maturation. Importantly, the improvement in binding affinity translated into a 70-fold improvement in neutralization potency against SARS-CoV-2 in a pseudovirus assay. After improving the potency of Adagio 2, we next wanted to confirm that it was capable of neutralizing authentic SARS-CoV-2, determine how it compared to other clinical stage antibodies, and ensure that it retained the breadth of neutralization we originally sought. We evaluated these questions through a series of authentic virus neutralization assays against SARS-CoV-2 and additional SARS-related virus, and this data is shown in the next slide. On the left, I'm showing a phylogenetic tree of some SARS-related viruses, highlighting the clade 1 viruses, which are of greatest interest as these are the group capable of infecting human cells via the ACE2 receptor. Of these, 4 viruses are available for authentic virus neutralization testing: the original SARS-CoV, SARS-CoV-2, and 2 bat viruses, WIV1 and SHC-014. For benchmarking purposes, we included the following comparator antibodies: VIR-7831, the 2 Regeneron antibodies, the 2 Lilly antibodies, and 1 of the AstraZeneca antibodies. As you can see on the right, Adagio 2 was as potent as, if not more potent, than the other antibodies tested against authentic SARS-CoV-2, with an IC50 value of approximately 1 nanogram per mil in 2 authentic virus neutralization assays using HeLa ACE2 and Viro target cells. In terms of breadth, Adagio 2 was the only molecule to maintain potent neutralizing activity against the additional SARS-like viruses tested, with IC50 values less than 10 nanograms per mil across these SARS-related viruses. None of the other antibodies tested had activity against these additional viruses, with the exception of VIR-7831, which is the only other antibody tested that was also isolated from an original SARS survivor. However, as you can see, VIR-7831 was approximately 10-fold less potent than Adagio 2 across the additional viruses tested. We next sought to determine whether our antibodies maintain potent neutralizing activity against not only additional SARS-like viruses, but also against the emerging SARS-CoV-2 variants of concern. We evaluated the neutralization potency of Adagio 2 as well as Adagio 10 against the B117, B1351, and P1 variants in the same authentic virus neutralization assay that I mentioned earlier. You can see that both Adagio 20 and Adagio 10 maintain potent neutralizing activity against all of the variants tested with IC50 values of 11 nanograms per mil or less. As illustrated earlier, many, but not all, of the SARS-CoV-2 specific antibodies displayed significant loss of potency against a subset of these variants. We've subsequently extended these findings. Extended these findings against a broad panel of pseudoviruses available through the Preclinical Services Program of the National Institutes of Allergy and Infectious Diseases. This panel includes single-point mutations, as well as additional variants of interest or concern, such as the B1.429 variant or epsilon, which is currently circulating in the Western United States. Across this broad panel of pseudovirus variants, Adagio 20 and Adagio 10 maintain potent neutralizing activity, with IC50 values remaining within 0.4 to 4-fold of the reference D614G strain used in this testing. The ability of Adagio 20 to maintain potent neutralizing activity against emerging SARS-CoV-2 variants resistant to other antibody-based therapeutics, as well as against additional SARS-like viruses, is explained by the finding that Adagio 20 binds to a unique, highly conserved epitope in the receptor binding domain of the spike protein that is not impacted by the mutations that lead to loss of activity of the common class 1 and 2 antibodies. We next sought to determine if the broad neutralizing in vitro activity of Adagio 2 translated into broad protection from infection in vivo. For these experiments, we used a mouse model of infection for SARS-CoV-2 and the original SARS virus, SARS-CoV-1. In these experiments, mice were administered 200 micrograms of Adagio 2 or placebo by intraperitoneal injection and were subsequently challenged 12 hours later with 10 to the 3 plaque-forming units of mouse-adapted SARS-CoV-2 by the intranasal route. The mice were then monitored daily for weight loss and respiratory changes, and then sacrificed on day 4 to evaluate lung, viral loads, and histopathology. Compared to sham-treated animals, mice treated with Adagio 2 showed minimal weight loss and preserved respiratory function and were completely protected against pulmonary hemorrhage and viral replication in the lung. We then repeated these studies with SARS-CoV-1. You can see the results looked almost identical, with minimal weight loss, preserved respiratory function, and complete protection against pulmonary hemorrhage and viral replication in the lung. Taken together, these two studies illustrate that breadth of neutralization in vitro translated into breadth of protection in vivo. In terms of our clinical program, we believe that intervention with an antiviral neutralizing antibody early in the course of infection and prior to the onset of significant inflammatory pathology is likely to provide the greatest benefit to patients. For this reason, our clinical development program for Adagio 20 is focused on the patient population shown in the box cells on the left, with the goals of preventing infection and disease in uninfected individuals and preventing disease progression by early treatment of ambulatory patients with mild to moderate COVID-19. Despite several emergency use authorizations for vaccines and monoclonal antibodies, we see 3 key areas of unmet need that our program has the potential to address. The first we've already discussed in great detail, and that is the need for agents such as Adagio 20 and 10 that have the potential to retain reliable activity against current and future SARS-CoV-2 variants, as well as additional SARS-like viruses with pandemic potential. The inconvenient IV administration modality for antibodies currently available under EUA has been a clear barrier to access for these otherwise potentially life-saving therapies. This issue illustrates the need for treatment options that can be conveniently administered across a wide variety of outpatient settings. With its convenient IM administration modality, Adagio 20 has the potential to address this need. Finally, there is an urgent need for alternatives or supplements to vaccines for populations such as the immune-compromised who are unlikely to generate protective immune responses to vaccination. With its extended half-life and potential to confer durable protection from COVID-19, Adagio 20 has the potential to fill this gap for these most vulnerable patients. Our clinical development program is designed to support the use of Adagio 20 to address these unmet medical needs. We currently have 3 clinical trials planned or ongoing for 2021. I'm delighted to report that we have completed enrollment of our first-in-human study with preliminary safety, tolerability, and PK data supportive of continued development. Critically, data from this initial study supported the anticipated half-life extension, enabling the potential for a single IM injection of Adagio 20 to provide protection from COVID-19 for up to 12 months. Based on these data, we are conducting 2 global, seamless phase 2-3 pivotal trials. The first is STAMP, a placebo-controlled trial in ambulatory patients with early, mild to moderate COVID-19 who are at high risk of disease progression due to age or comorbidities. The second trial, EVADE, is a platform trial designed to assess the safety and efficacy of Adagio 20 in the prevention of COVID-19 in 2 settings, pre- and post-exposure prophylaxis. In both studies, Adagio 20 is administered via intramuscular injection, and we will be targeting enrollment in regions with high prevalence of key variants of concern. In summary, we believe that our broadly neutralizing antibody program has the potential to address not only the current SARS-CoV-2 pandemic, but also future beta coronavirus threats. Adagio 20's unique combination of potency, breadth, convenient IM administration, and extended duration of effect support its potential use in the treatment and prevention of COVID-19, including diseases due to SARS-CoV-2 variants resistant to other antibody-based therapies. The potential combination of Adagio 20 with a second potent and broadly neutralizing antibody, namely Adagio 10, is designed to ensure durable efficacy against COVID-19 as SARS-CoV-2 variants continue to emerge, as well as against future outbreaks of disease that may arise from additional SARS-like viruses with pandemic potential. I would like to end by acknowledging the tremendous contributions of the colleagues and collaborators listed on this slide. Thank you for your attention, and I'm happy to answer questions.
Thank you very much, Lynn. That was very exciting, the results that you presented. There's 2 questions so far. The first has to do with how readily can you upscale production of this antibody for global access?
Sure. No, that's an excellent question, and it's an issue that's often been discussed around monoclonal antibody products in particular. And I'm happy to share that we have a very standard manufacturing process that is a very high titer, high yield process, which enables us to keep our cost of goods low, which we anticipate will enable very affordable pricing ultimately for our products for use on a global scale.
Okay. The other question had to do with, did you guys consider a pool of maybe 2, 3, 4 monoclonals where you could actually select them based on if a mutation did arise to one of them, it was incompatible with other mutations, that the function of the protein just is basically lost if you became resistant to more than one?
Sure. Yeah. So we initially started with Adagio 2, now Adagio 20, based on its highly conserved epitope and broad neutralization potency. With the anticipation that escape would be unlikely given the highly conserved nature of the epitope. Although we certainly acknowledge that these viruses, have tremendous capacity for mutation, which is the reason why we have additional broadly neutralizing antibodies such as Adagio 10, in our portfolio that target different mutations or different epitopes than Adagio 20 such that viral escape from one of the antibodies would be covered by the other antibody in that combination. So yes, we are considering for future development these types of combination antibody products for exactly this reason.
Okay. Have you seen any evidence that two mutations are incompatible with function? That the virus is now the function of the protein is lost completely if there's too many mutations in it, in terms of the neutralization of the antibody?
Sure. I mean, I think it's an interesting question, and I'm considering how we would sort of address that or how we would try and determine that in vitro. What we can do is look for viral escape, with serial passage. And what we know is that for these broadly neutralizing antibodies, we see far fewer escape pathways than for antibodies that are, targeted to these very common types of epitopes where multiple escape pathways are possible. And so we do think that there's limited capacity for escape from these, from the epitopes targeted by our broadly neutralizing antibodies. And perhaps this is due to the fact that some of these escapes lead to also impact binding to ACE2 and thus render the virus incapable of subsequent infection of human cells. So in essence, I guess that answers the question to say yes, we do believe that trying to force escape may lead to viruses that are no longer viable. Mm-hmm.
Okay. Thank you very much.
There are additional questions? Are there additional questions arising?
Hey, can you guys hear me now? Sorry.
Yep, you're back.
Yeah, sorry I don't know what happened. I don't see any other questions right now. So I think
I
we could
see
move
one
on to the
question
next
in
speaker.
here around whether. Okay.